Hello and welcome back to Excel Sheet Academy. So we were doing RNA splicing, and let us just summarize first like what we have discussed in the previous class. Okay, so we have talked about the different mechanism, that is like spliceosome machinery we have mentioned. Okay, so here like Akshay, you were not there. We'll just summarize for you today. So what we have discussed yesterday was that in order to carry out the RNA splicing, so if you remember, we have also mentioned about the 5' site and about the A branch site is there. GU is the sequences which is present at the 5' site. A is present at the branch site, and then AG is present at the 3' site. Okay. So in order to carry out the splicing, so this is a part of exon, this is a part of exon. Here you have got your intron present. So we need to remove this interon. Now we have got this spliceosome machinery. So this machinery over here, it has got several proteins which is involved in the splicing. Okay. So first one is that to the 5' splice, right, your U1 comes. Okay. So all these are your small nuclear ribonuclear proteins are there, in short known as NURBs. Okay. So there are 5 are there. 5 are there. Nomenclature is given as U1, U2, 4, 5, and 6. Okay, so this is how it is given. Now, so at the 5' splice side, U1 comes and interacts. At the branch side, BBP comes. So now what is BBP? It is your branch side or branch point binding protein. And to the 3' side, U2 AF 6535 comes. Okay, so 65 is the largest subunit which is interacting with the branch side. 35 is the smaller subunit which interacts with the 3' splice side. Okay, now once the BPP, that is, it makes sure that the A which is present at the branch side is protruded out so that it is available for the reactions to carry out. That is like the breaking of the phosphodiester bond, etc. Okay, so once this is done, BBP leaves and U2 comes and interacts. Later, this is going to displace and the combination of 4, 5, and 6 will come. This is finally going to displace U1 and there will be a cut in the exon intron site over here. So this is a first breaking of the phosphodiester bond. Okay. Then it is going to act over here at the branch site. So here the 2-OH, okay, 2-OH is going to have a nucleophilic attack on the phosphate present over here and forms a lariat. So your intron forms a lariat, not N, lariat. Okay. Later, the 3-OH, which is present over here of the exon, will have the nucleophilic attack of phosphate present over here. So resulting in the joining of the 2 exons and the removal of your intron lariat along with the other spliceosome machinery. So later on, this will get disassembled and the introns will be chopped off or it will be degraded by the RNases. So this is how the spliceosome machinery works. Okay, these are the steps involved in the removal of the introns. So there was this video which I wanted to share. So we can watch it again. Please watch this. If any doubts are there, let me know. Okay.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay. Akshay, got the idea?
Yes, ma'am. After the splicing, are the introns getting degraded?
Yes.
Okay.
So introns are nothing but your nucleotides only, right? So they will be degraded to the nucleotides, which will be later used for the further transcription. Okay. So nothing gets wasted. Okay. So now let's move ahead now. So the self-splicing introns reveal that RNA can catalyze RNA splicing. So these are the different types of the intron splicing mechanism where RNA itself will be catalyzing its removal. Okay. तो अभी हमने क्या देखा? It was using the help of the spliceosome machinery. ठीक है? अभी क्या है? They themselves can catalyze the RNA splicing. So this is the like 3 classes of RNA splicing where we have discussed. Okay, तो पहला हो गया अपन nuclear pre-mRNA. So where normally the mRNA is being transcribed. Okay, and the mechanism is to trans esterification reactions. Branch A site is there, and this is done by the spliceosome. Just now what we have seen. Okay, now we have got the other 2 mechanism that is belonging to group 2 introns and group 1 introns. So they are rare, and in only few eukaryotic genes it is present. Maybe in the organelles or in the case of the prokaryotes, rarely it is present. Now in this case, group 2, the mechanism is same as that of the pre-mRNA, but instead of spliceosomes, we have got the ribozyme. Okay, same. Now coming to the group 1, it is again the 2 trans esterification reaction. Instead of branch A site, here in the case of group 1, we have got G, and the mechanism or the catalytic machinery is same as your group 1. यानी कि दोनों ही हमारे ribozyme हैं. Okay, so this table you need to understand in such a way that so you can understand किसमें क्या similarities है. So if you see normally nuclear pre-mRNA and group 2 introns, the mechanism is same. Okay, I mean mechanism is same for all. Only difference is that here the branch site is A only, but in the case of group 1 we have got G. Okay. Now the catalytic machinery, like how it is being carried out, it is with the spliceosomes, whereas in the case of the introns, group 1 and group 2, it is because of the ribozymes. Clear? So just go through once and you'll be able to understand. So now I have got the diagram which will make the what to say the confusion dur karega. Now first let's look at the what you say, let's look at the spliceosome wala only. So we know that you have got the branch site A which shows the nucleophilic attack, and यहाँ से attack होगा, यहाँ पे आपका branch बनेगा, right? And finally this will have attack over here, and finally the exons are joined and the introns are removed. Okay. Now, if you could see that over here in the case of group 2 self-splicing, you don't need any machinery. It itself is acting as a enzyme. So A, again it is present as at the branch site. So this 2-OH nucleophilic attack over here, because of which what will happen? This will get joined. Okay. So here also you can see that it is forming a lariat. Okay. It is also forming a lariat over here. Then 3-OH is attacking at the phosphate group present over here. Similarly, the 2 exons are joined and the lariat is removed. Whereas in the case of group 1 now, in the case of group 1, instead of A, you have got G present. Instead of 2-, you have got 3-. Can you see the difference? Okay. So ये क्या करेगा यहाँ पे, इसको attack करेगा. But what is happening? This G will get inserted over here. यहाँ से हटके वो यहाँ आ जाएगा. Okay. And it is removed as a what to say, a linear intron. It is not forming a lariat. Can you see? Ultimately, the exons are joined, okay, fused, but your introns is not forming a lariat. Instead, it is forming a linear intron, and this will be degraded further. But both these two, okay, these two scenario, group 1 and group 2, it is the example of ribozymes. Clear? Tanvi, it is clear?
Yes, ma'am.
Okay, it's a very simple concept. Okay. Now this is how they are showing ki See, when we saw in the case of the pre-mRNA, the U2, U6, those machinery were forming the complementary base pair with your introns. Okay, so this is how it was able to have the catalytic reactions. Okay, for example, let us look at over here. So we had A2-OH, which was acting over here and joining and forming a lariat, right? And we have the complementary base pairing between the U2 and the introns. Similarly, in the case of the ribozyme, you can see that it itself forms a base pairing. Okay, so those are known as domain 5 and domain 6. So ये आपका इंट्रॉन ही है जो एक दूसरे के साथ बेस पेयर करके हेल्प कर रहा है सेल्फ स्प्लाइसिंग में. So it is just mimicking the activity of your spliceosome. Can you see? Can you correlate? So जहाँ पे U6 is making interaction with U2, similarly the domain 5 and domain 6 are making interactions with the intron. तो ये आपका ये domain 6 है, ये आपका intron का part है, which is making a complementary base pairing. Finally, the A का 2-OH is attacking at the GU of the intron. Okay, so it is itself, okay, the RNA itself is acting as the enzyme, mimicking the activity of the spliceosome and hence resulting in the RNA splicing. Okay, so it is the mechanism this which is required for by you guys to understand. Okay, diagram to koi nahi puchega, but please do understand that if the question comes like who is mimicking what and all, so just remember U6, U2 are mimicked by the domain 5 and 6 of the group 2 RNA self-splicing introns. Okay, now the question comes. So that was all about the RNA self-splicing. Now let's talk about the spliceosome itself. How does the spliceosome make sure that these are the correct site at which I need to carry out the splicing, right? So we need some proof, we need some authentication by which the spliceosomes can make sure that जो भी हम splice करने जा रहे हैं, those are the correct regions. Okay, so everything needs to be foolproof, checkpoint होने चाहिए. So here also, so they prevent the use of incorrect sites by ensuring that the splice sites are close to the exons and are recognized preferentially. अब ये कैसे करते हैं? So they have got a different set of proteins. Okay, those are known as the SR proteins, which is standing for serine arginine rich proteins. Okay, so they go and bind to the regions called exonic splicing enhancers. Okay, they are present in the exons itself. So till now what we have seen कि हमारे पास यहाँ पे अभी exon है, इतना portion intron है, let us say. Okay, then again we have got exon. So these ESEs, that is exonic splicing enhancers, are present in your exons region, correct? So to this your SR will come and bind, those are your serine arginine rich proteins, and will make sure that the splice sites are near to them and which will make it efficient for them to bind. Okay, so these SR proteins will recruit all your spliceosome machinery, for example, U2AF proteins and U1 at the 5' site and U2AF at the 3' site. Okay, so this recruitment is either through the direct interactions between the SR proteins and the spliceosome proteins, or it is with the RNA-RNA hybrid which is formed during the spliceosome assembly. So let's see how these SR proteins are helping. Look in the figure. Now you can see that we have got the exons and introns. Okay. Now, so what do we see over here? In the exonic region itself, you have got the exonic splicing enhancers units are present, right? So to this only the SR protein, serine arginine rich proteins are binding. Now, can you see that very nicely they're interacting with the U1 also as well as A35 also, correct? And we know that this region binds at the 3' splice site of the intron, and U1 binds at the 5' splice site of the intron. Clear? So all these proteins are making sure that they are present correctly. The spliceosome machinery are present correctly and will carry out the further steps. Is this slide clear? Yes, Tanvi, Akshaya?
Yes, ma'am.
Okay. Good. So this is how the spliceosome machinery is making sure that whatever regions they are going to remove are correct. Okay, they are authentic regions to be removed. Now There is another type of splicing which in few cases of viruses and the other organisms it has been observed. So it is known as trans splicing. अब नाम से ही पता चल रहा है that it is trans यानी कि different it is. Okay. Now see, you have got RNA1 and RNA2. So what they are showing over here is that again, it is the branch side and the GUAG sites are there, which is normally present in the introns, जो अभी तक हम पढ़ते आ रहे हैं, correct? The only difference is that ये exon 1 का intron है and ये exon 2 का intron है. So these are getting joined or fused, and later यहाँ का जो 3-OH आएगा after making a cut and all, this will go and act over here. So those exons are getting fused. So what is happening? Two different exons are getting fused. समझ में आ रहा है? This is RNA1. This is totally separate mRNA. This is totally separate mRNA. The two different mRNAs are present. Their introns are getting fused and their exons are getting fused. Now, what kind of shape is this? So when they showed like this, they call it as lariat. Now, what kind of shape is this for you guys? कौन सा लेटर लगता है आपको?
Y.
Yes.
Y.
So it is also known as Y-shaped splicing. ठीक है? If in the exam they are not mentioning trans splicing, then they will mention as Y-shaped splicing. तब आपको आंसर क्या देना है? That the splicing is happening between two different mRNAs or RNAs. Okay, clear? तो अब तक हम क्या डिस्कस कर रहे थे कि विद इन द सेम mRNA the exons are getting fused. Here it is different RNA, इसका exon and इसका exon साथ में आके fuse हो रहे हैं and these introns are getting fused in the shape of the Y. Okay, clear? Simple topic. It's just that 2 marks के लिए एक बार ये question आया था. That's all. Very simple. Excuse me. Okay. Now let's come to the alternate splicing. Okay, so alternate splicing. Alternative splicing itself is telling you like what kind of splicing it is going to result. So guys, if you remember, okay, so we have discussed about this when we were studying about the RNA splicing. If you remember, we discussed about the VDJ recombination. I have mentioned about this kind of recombination VDJ, which results in increasing the diversity in the case of your antibodies, if you remember. So this is for your variable diversity in joining genes. So alternate splicing says that it is the same gene, it will give you the transcript, and that transcript, depending on the differences in the splicing, can result in the production of different types of proteins. Understood? Let's see the figure, that will be more clear. So if you see over here, we have got the primary RNA transcript. So we have got exon 1, 2, 3, 4, and 5 are there. This light green color things are your introns, correct? Okay. Now, if the splicing happens and I got the sequences 1, 2, 4, and 5. So there is no exon 3 taken. Okay. This results in the formation of splice mRNA beta troponin capital T. Okay. Now, if suppose the splicing happens in such a way that 1, 2, 3, no 4, and 5 has been taken, then it results in the formation of your alpha troponin T. So it is the same transcript which will result in the different forms of your protein. So alpha, beta, just 1 or 2 domains are here and there because of the alternative splicing. It will give you different isoforms of the protein. Clear? So if you see here, it is alpha, this is beta. So just because of the just because of the differences in the exons rearrangement, you are getting different expressions of the proteins. Clear? Now, we can, this will be more clear as we see more examples. Now, it is not that like always 1, 2, 3 and 1, 3 or 1, 2 only you will get. So there are many ways by which the RNA can get spliced. So here they have shown 5 mechanisms by which the RNA splicing can be done. So now you know the mechanism behind the splicing. Either it can be your spliceosome और ग्रुप 2 ग्रुप 1 इंट्रॉन आरएनए स्प्लाइसिंग, ठीक है, वो एक डिफरेंट सिनेरियो गया. नाउ व्हाट इज द एंड प्रोडक्ट ऑफ दिस splicing? So those can vary from organism to organism. For example, either you can have normally, so let us consider that we have got here 3 exons are there, okay, and we have got 2 introns. Okay, let us see this example. So you got the primary transcript also. Now, if the splicing happens normally, then you should get a normal splice mRNA where it has got all the 3 exons, correct? Now, if I get only 1, 3, it is known as exon got skipped. Okay. If I have 1, 2, 3, but can you see what this portion indicates? From this, what you will understand? If this portion has been included, what does it mean?
Intron part is there.
Yes, correct. So when someone says in literature or when you read a research publication, if they say that the exon extended primary mRNA transcript it is, then it means that extension means that the entire intron is not being removed. Okay, so it is present over there. Now, intron retained, what does it mean? The entire intron has been retained over here, correct? Now, over here, look at see. So either you can have 1, 3 or 1, 2. So these are alternative exons, right? The combination. So यहाँ पे क्या था? यहाँ पे हमने एक exon skip कर दिया. So you will have 1, 3. We can also have 1, 2. I'm skipping the third one. So these are the different forms by which you can get the exons. So you have seen that it is not necessary that every time the complete intron is being removed. Either a part of the intron will be retained or the entire intron will be retained. Okay? Not necessary that only this intron. ऐसा भी हो सकता है that you can get 1, 2 and little portion of this intron present over here and then 3. So if you ask why this is happening, it's all in order to create diversity in the organism, okay, in the species. So all these are going to result in some form of differences in the protein structure. So even though it is the same gene, it is the same primary RNA transcript, but once it gets expressed at the translation level, you are going to have different forms of the protein having different functions. And this is going to vary from organism to organism. Okay. Is this much clear? So we have got one example also. Yes. So the example is of about the monkey Sivian virus. Okay, 40. So it has got T, capital T antigen and small t antigens are there. So these are the main 2 antigen which is encoded by this virus. Okay. So the 2 proteins result from the alternative splicing of the pre-mRNAs from the same gene. So now let's see. So both, of course, the capital T and small t antigen, they have got the different functions. So large T induces transformation and cell cycle re-entry. Iska matlab kya? If you have got infection by the large T antigen, then the cell is undergoing to become cancerous. Okay. Now, the small t blocks the apoptotic response of the cells forced on that path. So the ratio of the 2 which is formed differ depending on the level of the splicing regulator SF2 and ASF. So which is your splicing regulator in the case of the Sivian virus 40? It is SF2 ASF. Okay. ऐसे याद करने की जरूरत नहीं है. It just for you to for you guys to understand only. Now, when these are present at the high level, the protein directs the machinery to favor the use of the 5 dash. It is 5 dash, guys. Okay, it's not 50. It is 5 dash splice site that generates more of the T antigen mRNA. Okay. अब इसे क्या समझ में आ रहा है? If I have got more of 5 dash splice site, then it is T antigen. Now, SF2ASF is an SR protein, and when it is more, it binds sites within the exon 2 and helps the splices to assemble there. Okay. Now, these together, the binding of the SR on the exons, so we know that the exons bind to the ESC regions within the exons. So all these are known as the cassets. Okay, exon cassets. Ye kuch nahi hai. Exon cassets. Okay. So what is the takeaway message over here? That we have got the T antigen and small t antigen. this is responsible for transforming the cell and the cell cycle re-entry. यानी कि जो stop हो जाना चाहिए था आपका mitosis के बाद, it is not stopping, it is just continuing. Okay, and small t antigen is blocking your apoptosis. Now this is all depending on like whether the expression of the mRNA should be giving you capital T or small t. It depends on the splicing regulator. Okay, now let's see, the figure is there which will make it clear. Okay, so here we have got the primary transcript. Okay, so let's see, the primary transcript is present. Now if you see, we have got the stop codon present over here. Okay, so now when we have got this, okay, the small t antigen is encoded by the longer of the two mRNAs. Small t is encoded by the longer of the two mRNAs over here. Okay, so now if suppose here, if it is containing the mRNA, okay, if you see, this much of the intronic region has been retained in the mRNA. When it is translating, what will happen? You have got the stop codon here itself, so the protein which will be synthesized will be of this much only. Are you getting point? Yes.
Repeat.
Okay, I'll repeat once again. So what are these SST and all? We'll talk about that later. Okay, so you have got the exon 1 stop codon sequences are over here, and then you have got the exon 2. Now you got your primary primary RNA transcript. So where it has got one, the intron region, and your second exon region. This is your DNA. Now you got your RNA. Now when I say mRNA, it is your spliced mRNA. Okay, this is primary without splicing. Only mRNA, when it is said, it is your spliced mRNA. Now, as we have seen earlier, after splicing, there are chances that you are going to retain the intron part. So, as you can see over here, the exon 1 is retained. One second. Exon 1 is retained, and you can also see a part of intron. A part of intron is also retained, and then exon 2 is there. Is this much is clear? Exon 1, a part of intron, and exon 2. Yeah. Now, when this is being translated, so what we have seen is that there is a presence of stop codon. अब stop codon यहाँ पे बीच में आ जाएगा, so obviously it will stop here itself, correct? So what we get is the small polypeptide chain. So if the splicing is in such a way that the introns is being retained, then you are going to get T antigen. Okay, abhi clear hai? Now, if the intron is completely removed and I get only exon 1 and 2, then I'm going to get is T antigen. Now it is clear? So retaining intron part is also a part of splicing mechanism by which there is a difference in the expression of the proteins. So these antigens are nothing but proteins only. Now, depending on the signal, what they get, okay, the mRNA is going to get translated in such a way that either you're going to get small t antigen or you're going to get a capital T antigen. Okay, now it is clear. Now let us see what is these SST. Okay, so this SST refers to, agar main yahan pe bol rahi hu that it has got phi-SST, it refers to the phi-splice site. Okay, phi-splice site used to generate the larger tmRNAs. Agar main yahan bol rahi hu, okay, phi-small SST, so it is the phi-splice site for the small t. Okay, then we have got 3' site used in generating both mRNAs. Is it clear the concept of SST? Nahin? It just means that, see, agar mujhe capital T chahiye, so the capital SST, yahan pe mera wo sequence hai, so all my splices of machinery will come and sit over here to remove introns from here and here. Okay, clear? In the case of capital T. Now, but if I have to remove for my small t, then the splicing is happening between this and this. So only this much region is removed. Ab clear hai? Akshay, is it clear? Or should I repeat this SST wala part?
Yes.
Okay, I'll repeat. Okay, this much is clear to you that if the intron is being retained, a part of intron is being retained, then you get small t antigen. When everything is removed, the complete intron is removed, you get capital T antigen. Itna clear hai dono ko?
Yes, ma'am.
Yes, ma'am.
Okay. Now, what is not understood is this SST wala part. Okay. Now, SST ka matlab kya hai? It is your phi. See, when they say phi dash capital S T, it means splice. It is your splice site for your capital T antigen. Okay. So the splices of machinery will come and sit over here, make the interactions over here. Okay, if you remember your splices of machinery, U1 comes and sits over here, and to the branch side it comes and sit over there and makes the lariat and all, etc. Correct? So in the case of capital T antigen, the splices of machinery is going to sit over here and at 3-SST. Okay, so here U1 will be there, here your U2, AF, 65, 35 will come, and then they will remove the entire intron in the form of the lariat. This is just like normal splices of machinery between two exons. Is this much clear now? Abhi clear hai?
Yes, ma'am.
These are the regions where your splicing machinery is going to sit. Now, when you talk about your small SST, that is for phi dash splice site for your small t it is. Okay, it is for your small t. तो उसको हम क्या बोल रहे हैं? That splices of machinery should sit in such a way that the resulting in future I should get a small t antigen. So the splices of machinery कहाँ बैठेगा? यहाँ बैठेगा. So U1 यहाँ बैठेगा. एंड सिमिलरली U2 AF जो है आपका नॉर्मली वो आपका 3' साइड में ही बैठेगा. सो हाउ मच इंट्रॉन्स विल बी रिमूव्ड? So this much of the intron. So this much of the intron has been removed, and rest all is retained in your matured spliced mRNA. Abhi clear hai? So it is these sequences which is present, which is deciding that हाँ कि मेरे splices of machinery यहाँ आके बैठना चाहिए and इतना intron हटाना है. It is not a random chance. Okay, it is not happening randomly. It is all predetermined in the sequences. Now, do you understand like why introns are important? We do say that in our genome, so much of introns are there, but they are not junk. They do carry the information. They do decide what kind of proteins are needed to be expressed after translation. Abhi clear hai?
Yes.
Okay, both of you clear?
Yes, ma'am.
Okay, so it is these sequences which decide. So एक example था कि कैसे अगर हम intron retain कर रहे हैं mRNA में, so what kind of protein we can get? Without introns हम हमेशा ही पढ़ते हैं. We always say that without introns, exons are there, they will be converted into the protein after translation, right? But we also have introns carrying the information. Okay, okay. Now, so till now we discuss all about key splices of machinery kaisi hai. We also discuss about self-splicing introns. Okay. Now, there are certain enhancers. Okay, the activators and repressors are there. So as we discussed earlier that in the exonic splicing enhancers, for example, SR के बारे में हमने पढ़ा जहाँ पे serine, arginine rich proteins are coming and helping these splices of machinery in order to carry out the splicing. Correct. So we do also have the proteins which is going to help, like the rate of splicing they're going to increase, and we do also have proteins which is going to silence, like they are not telling you, they are telling you not to splice. Okay, so we have got the repressors as well as the enhancers. So if you see in this example, if you see, so we have got normal. This is your normal scenario where the splicing sites are there, repressor sites are there, splicing machinery will come and it will remove your introns and you will get the mRNA. Okay, spliced mRNA you will get. Now, if you have got this scenario where the repressor is present, okay, and it is telling you that we don't want splicing to happen, so what will happen? The repressor will go and bind. Now, since repressor is binding to that region, your splicing machinery cannot sit. Already वहां पे एक bind हुआ है, तो दूसरा आके नहीं बैठ सकता है. Now, this is only happens when the splicing site का information, that is sequence information, is overlapping your repressor का sequence information. So because of this, what will happen? अगर आपका splicing machinery बैठता है, then repressor cannot sit and splicing will happen. But if the repressor is sitting over here, then your splicing machinery cannot sit and the splicing mechanism will not happen. Clear? Simple. Repressor present है, splicing नहीं होगा. So what you are going to have? The unspliced mRNA. But again, this all information is provided कि मुझे splicing करना है या नहीं करना है, and depending on that you're going to have the protein expression. Now, second scenario where you have got splicing machinery and you have got the enhancers. So if it is sitting now in this case, if you don't have the enhancers, okay, if you don't have the enhancers, the splicing machinery cannot proceed with the splicing. So here the splicing machinery meets the requirement of the enhancer proteins. ठीक है? अब enhancer नहीं है, splicing नहीं होगा. Enhancer है, splicing होगा. Correct? So here you have got the enhancers or the activators. So until they come and activate your splices of machinery, it is not going to splice. So here you need the activator in order to have the spliced RNA. Here, if you have got the repressor, then you're going to have unspliced RNA. Clear? Am I going fast? Akshaya, are you able to follow? Simple. Or should I go a little slow?
It's fine, ma'am.
Okay. I mean, once you listen again, you will be able to understand. I mean, you will be able to remember also. Yeah. Simple. Okay. So we do have enhancers and the repressors for your spliceosome. So अभी तक हमने क्या देखा in the spliceosome? So we talked about the spliceosome machinery. Correct? We talked about self-splicing RNA. So where you need to talk about intron 1 and 2. Okay? Then we talked about, then what did we talk? Then we talked about the trans-splicing. Then next we talked about the alternative splicing, in which we discussed like how retaining the intron part or not retaining the introns will result in the expression of two different proteins. Let it be antigen, where we have seen the example of SB40. It is Josevian virus 40 found in the monkeys. Okay. So this is what we all discussed. Now, in order to enhance these splices of machinery, we have got the enhancers and the repressors. So if you have got the repressor present, no splicing will be happening. If you have enhancer, then only the splicing will happen. No enhancer, no splicing. No repressor, splicing is there. Okay, simple. Now, the another important topic is about the exon shuffling. So like how you have got the alternate splicing, which results in the formation of new protein expression, exon shuffling is also something like that, which will result in the formation of new genes. See, अब वहाँ पे क्या था 1, 2, 3 था, अगर 1, 3 हो जाएगा, so we are getting what? A different expression. So this protein expression is going to be different from this. Similarly, if all these genes are getting shuffled, like 1, 3, 2 हो जा रहा है, so I'm going to get a different itself, right? This is going to be new gene itself. So it is a molecular mechanism for the formation of new genes. It's a process through which two or more exons from different genes can be brought together. Or you can have the creating the new exon intron structure if you can duplicate the same exon. Suppose 1, 2, 2 kar de rahi ho. So 1, 2, 3, 1, 1, 2. So even though the number or the same exon it is, but if I have got these exon reshuffled, okay, the arrangement of these exons are different. Obviously, these are going to result in 3 different protein expression. Okay. Now let's see more in detail. So exon duplication. So you have got the parental chromosome, which is carrying all these exons 1, 2, 3. Okay. So you are having exon 2, exon 2, exon 3 is there. Now, if suppose recombination is happening and it is having the unequal crossing over, which is possible. So what will happen? So instead of having a complete recombination, you are going to have exon 1, 2, 3 and 3. So what is happening over here? Exon 3 is getting duplicated. Okay. So in future, when this is going to give the transcription, it is going to have 1, 2, 3 and 3. So this particular gene is going to be different now. And this particular chromosome is going to give you now exons 1 and 2. So the expression of this gene and expression of this gene is going to be different. Now, ye kyun hua? Because of the exon duplication. And this is how you bring about the diversity in the proteins. Okay. Now, this is exon duplication. What if the entire gene is getting duplicated? So here, in this example, this one thing was considered as one gene. ठीक है, gene के अंदर exon. ये हो गया आपका पहला example. Now let's talk about if the gene is getting duplicated. So if you have got the gene, this is the entire globin gene is there, and here you have got the another globin gene. Now because of again similar recombination which is happening unequal, you are going to have two genes over here in the same recombinant chromosome and the other is not having. Okay, so this will result in the gene duplication. This is just similar to your sorry, I should say in the other way around. So how the gene duplication happens, in the same way the exon duplication also happens. So this we'll study in much more detail when we'll start doing about the genetics. Okay. Similarly, the gene gets duplicated in the same way the exons are also getting duplicated just because of the unequal crossing over. Okay. Now let's see for a few more examples and it will be more clear. Now before we proceed, why introns are rare, almost non-existent in bacteria? Can you guess the answer? Why does a bacteria does not have introns? Is it like they got lost or it never existed? और ऐसा क्यों है कि यूकैरियोट्स में इतना सारा इंट्रॉन्स है? Yes, Tanvi, what do you think? What will be the answer for this? Any guesses?
To create diversity.
That is in the case of eukaryotes, right? If I am asking why introns are not present in the bacteria, then yes.
Ma'am, is it because there is no complex function in bacteria, and the size of DNA is also very completely small when
Yes.
compared to eukaryotes?
This is one correct point, that the size of the DNA is small as compared to the eukaryotes. Okay, so bacteria are under pressure that they have got a small genomic DNA and they need to they need to divide fast, because as a single bacteria they can't do anything, so they need to have more colonies. So there is a pressure on them because it is easy to destroy such a small living beings, right? So they are under pressure, and that's why they don't want to waste time in replicating or transcribing introns region. So there was a pressure on them for not having the introns and having only the functional regions in the DNA. Okay, now who is deciding all this? The evolution. Now why does it happen? Like, why in the case of eukaryotes we have got, so as the eukaryotes were growing, of course there was increase in the complexity in order to have more diversity. So diversity सबसे ज़्यादा कैसे आएगी? When you have got more introns. And how these introns number got increased? By mixing of the DNA. Many a time these eukaryotes were getting infected by these virus, so उनका genome वहाँ पे insert हो जा रहा था. By next time, even though if you're resistant to the viral attack, but the small part of the virus got integrated into your genome. So in the next round of replication, that has become now your genome part, okay, even though it is now known as introns. So this is why, and plus जैसे जैसे complexity ज्यादा है, the chances of having mistakes during the replication is more. So it's okay if the mistakes are happening in the intronic region. So the chances of having less mistakes in the exons regions are increased when there was increase in the intronic region. So these are the answers like evolutionists have thought about it by giving such answers. Okay. So this is just to think about it. Nothing like I'm sure nobody's gonna ask about this. Okay. So there are two models which talk about the exon shuffling. Intron's early model. So introns existed in all organisms but have been lost from the bacteria just because of the natural selective pressure was there. So they became good gene-rich organism. Or then we have got the another model which says that intron late model. So introns never existed in bacteria but arose later in the evolution. So as the diversity was increasing, the evolution has started adding the introns in the genome of the eukaryotes. Okay, so these are the models. As I said before, also models are something which the scientists or the researchers have hypothesized. Okay, we don't have as a hardcore experimental evidence to prove it. Okay, okay. So these are the methods. Now, what are the advantages of having the presence of the introns? The presence of introns and the need to remove them. Okay, it gives an opportunity of the alternate splicing, which can generate multiple proteins now from a single gene. So how we have seen just now, right? From the single gene, from the single pre-mRNA transcript, now because of the presence of the introns, the exon shuffling can be done, the alternate splicing can be done, right? Now you can have different isoforms of the protein. Secondly, having the coding sequences of genes divided into several exons allow genes for the reshuffling exons, same thing. Now, what are the evidence which proves that that these things are present? Okay, so the borders between the exons and introns within a given gene often coincide with the boundaries between the domains within the protein encoded that gene. Acha. So for this, you need to have more of the structural knowledge. So if you see the structure of the proteins where these domains are present, every time whenever you see any protein domain region is present, always the start and end of the domain, there you're going to have the intronic region also. Okay, so that has proved that the introns are always present, exons, introns are always present. Okay, clear? Clear? So they have got this मैं थोड़ा diagram बना के समझाती हूँ. I hope I will be able to show. So let us say that this is exon 1, intron 1, exon 2. Okay. Now let us say that this is coding for one protein domain and this is coding for another protein domain. Now what we have seen is that at the border, ये जो sequence रहता है, ठीक है, at the border of the exon and intron, the sequences, many a times, as we know during the alternate splicing, many a times the part of the introns are also retained in the primary transcript, pre-mRNA transcript, correct? So this intronic region can also be seen at the C terminal of your protein domain. Okay, so उसका कुछ काम नहीं है, but since in order to get a proper structure of the domain, it is present. अभी थोड़ा सा समझा? Yes? Should I repeat?
Yes, ma'am.
Okay, okay. So many genes and the proteins they encode, they have got the, they have apparently arisen during evolution as part of exon duplication and divergence, what we have already seen. Okay. So these are the people who says that, you know, कि अगर हम domain structure properly analyze करेंगे, so we can see that certain intronic sequences are also present. That's all. Now the main part, this is the last topic of RNA splicing and the important one. जितने भी आज तक कोई अगर क्वेश्चन आया है, as I mentioned before also, mainly they will ask from your splices of machinery. Okay, they will, or the second important topic is RNA editing. So as now you can see, we have discussed कि कैसे हम किसी भी जीन प्रोडक्ट को हम चेंज कर सकते हैं with the help of the alternate splicing or exon shuffling. ठीक है, so RNA editing is your third mechanism by which you can change the sequence of your mRNA. So these are the three mechanisms by which the sequence of the mRNA will be changed before translation, and then only it enters your translation stage. Okay, so I believe that now alternate splicing, exon shuffling is clear to you guys. Clear? Can I say at least the concept is clear? Yeah. Now let's talk about this third mechanism, that is RNA editing. So here also we have got two ways by which RNA editing can be done. अब यहाँ पे क्या हो रहा है? Let us assume that the alternate splicing or exon shuffling के बाद you got your mRNA. ठीक है? Now again there was a sequence change required. Then you are going to edit the RNA. Now when you say edit the RNA, it's like literally you are inserting or changing the nucleotide sequences. For example, before I proceed, I'll just say one example. So let us say that you have got the CC sequence is present as a codon, right? So, but later the sequence, later the information was given to the cell is that this particular thing should be UAA. Okay. Now, what is happening? Cytosine से uracil बन रहा है. So, there is a deamination. Okay, deamination procedure happening. So, cytosine में अगर आप amino group हटा दोगे, so that will become uracil. Now, you tell me what is UAA coding for?
Stop codon.
Correct. So, if I have got the RNA, उसमें यहाँ पे CAA था, let it code for something. I don't remember. It is coding for something. But if I go and edit, like I make sure that the C में से मैंने amino group हटा दिया, C becomes UAA. But once this is going to translate, it will get stopped here itself. Correct? So now this mRNA is synthesizing or giving information or sorry, getting translated into some other protein जो इतना बड़ा बनना चाहिए था, वो इतना छोटा ही बना. So this is one way by which you can change the fate of the mRNA. Okay. And the second mechanism is by which there is a guided RNA. Okay, guided RNA. It is also known as gRNA. They go and help in inserting the certain nucleotides in between of these sequences. Okay, so they break the phosphodiester bond between the RNA, mRNA, insert the new sequences and reseal it with the help of the RNA ligase. तो जिससे क्या होगा, आपने नया सीक्वेंस ऐड किया है, so obviously नए न्यूक्लियोटाइड्स ऐड किए हैं, so obviously the sequence is going to change and now that particular mRNA is going to code for something else. Okay, इतना क्लियर हो गया है अक्षय, तन्वी, like how these two are there. A क्या करता है, C को सीधा A करता है by having the deamination reaction. A क्या करता है, it just goes and base pair with the RNA and break the phosphodiester bond between the RNA and insert new nucleotides and reseal it. Okay, so these are the two ways by which it is doing. So now let's see in detail how this mechanism is being achieved. Okay, so RNA editing is another way of changing the sequence of an mRNA. It is another mechanism that allows RNA to be changed after transcription to encode different protein. Okay, encoded by the gene. So there are two methods, that is site specific deamination, guide RNA directed uridine insertion or deletion. So अभी क्या हो रहा है, site specific deamination. So जैसे कि मैंने बताया CAA आपका UAA बन रहा है, so stop codon आ जाएगा, it is going to code something else. Now guide RNA or gRNA, it is directing the insertion of the uridine or deletion of the uridine. Okay, तो U जो uracil group है वो आपका either insert होगा या delete होगा. Now let's see more. Let's take an example now. It is always better to have an example for better understanding. So this is a tissue specific manner by deamination of a specific cytidine to generate a uridine. So C से हम U में जा रहे हैं and the enzyme is known as cytidine deaminase. Okay. So what do we see over here? So there is a codon of 2153, okay, and at the exon number 26 of the pre-mRNA, what is happening? You have got the spliced mRNA. आपने सारे introns हटा दिए, okay. Now this is your spliced version. Now, if this is encoded into the liver, okay, there is no editing. So this particular mRNA is going to get translated to a protein which is having 4563 amino acid protein. Okay, so it means CAA है जो आपका glutamine code करता है. Now let us say that this particular mRNA was transcribed and it is in the intestine. So it is happening over there. The site-specific deamination is happening there. Your cytidine deaminase is activated and it converts your CAA codon to UAA, which is a stop codon. So what will happen? It will just code till here. And that will be your around 2153 amino acid protein. Okay, clear? So depending on the tissue specificity, the site specific deamination will be carried out in order to get different expression of the protein, but from the same gene. It's the same gene giving you different protein function or protein expression. Okay, clear? Is this example is clear?
Yes, ma'am.
Okay. Now, there is another protein which can do the same. Okay, तो हमें मालूम है cytosine में भी amino group है, and we also have got the adenosine having the amino group. So in the case of cytosine, there was cytidine deaminase which was removing the amino group from the cytosine to get uracil. Now another enzyme is called ADAR. This is important. ये exam में आ चुका है. Okay, ADAR, which stands for adenosine deaminase, which is acting on the RNA. Adenosine deaminase acting on the RNA. So what it is doing? It is removing the amino group from the adenosine. Now, once the amino group is removed from the adenosine, it forms inosine. Okay. Later, this inosine can base pair with the cytosine, and overall, the entire mRNA sequence will get changed. So how they are doing, that is also shown in the figure. So you can have cytosine, amino group is removed, it becomes uracil. Now, if you have got the adenine, then the amino group is removed, it becomes inosine. Clear? So this is one way by which the site-specific deamination can happen. Now let's talk about the guide RNA directed uridine insertion or deletions. Okay, so here multiple U's can be inserted or it can be deleted. Now this kind of RNA editing happened in the mitochondria of the trypanosomes. What are trypanosomes? Yes. What are trypanosomes?
Protozoans.
Huh? They are protozoans, correct? They are the parasitic protozoans. Very good. Okay, so they have got this kind of RNA editing. Now, so U's are inserted into the message by the guide RNA. Let's see. So let us say that this is your unedited RNA. This is your mRNA. ठीक है? Now, इस mRNA में अभी आपका extra U add होगा. So how it is done? So this guide RNA is going to have the poly U stretch. Okay. Then the editing region, which is going to base pair with this. So you can see GC base pair हो रहा है, right? So जहां जहां base pairing नहीं हो रहा है वहां पे वो insert करेगा भी. Now the anchor directing the guide RNA to the region of the mRNAs, it is going to edit. Now when you say anchor, इसका मतलब क्या है? There has to be a proper base pairing then. You can see AU, GC, CG. So here you can have the proper, sorry, one more is there. Proper base pairing happening, correct? Then you have got the poly U stretches there. Now this is the region where now they're going to add the U. Okay. Now this is very crucial to understand. Let's go slowly. Okay. Yeah. So the first scenario, okay, what do we see? We have got the DNA, we have got the RNA, and the extra U has been inserted, because of which the protein expression is changed. Okay, now how it is being done? Let's see. One second. Okay, now look at over here. So you have got the mRNA, you have got the gRNA. Can you see it? Now these are the site where the U are going to get inserted. Now how it is being done? how it is being done. Can you see? They make a base pairing over here. Now, wherever it is not matching properly, okay, see, यहाँ पे उन्होंने extra add कर दिया है, endonuclease is there. तो जहां जहां mismatch होगा वहां वहां वो they will make a cut. Now, when I say cut, what does it mean? They are breaking the phosphodiester bond of the RNA. They are going to add U. So UTPs will be added. Okay, of course, I need RNTPs, right? So UTP will be added, and that is complementary to your RNA ka. gRNA. So gRNA is going to have AA. So uske opposite mein you are going to add U. Okay, can you see it? They have added U. And later this gap, okay, this gap is present, right? So that will be joined by the RNA ligase at the end. Clear? So what you need to remember is, in the case of the gRNA editing, that is guide RNA editing, they add a stretch of U. And now that is done by the sequence followed onto the gRNA having poly U stretch should be there. Okay, they need to have the editing region and the anchor region. So once it is being formed properly, the anchor region is formed, okay, they base pair properly, then the endonuclease will come. It is going to make a phosphodiester break. Then the extra U's will be added opposite to the A, which is present onto the gRNA ka sequence. Then what is happening? The RNA ligase will come and it is going to reseal. And this is how the extra U's are added into the sequences. So at the end, what is happening? You are going to get a sequence where GA, UU, GU, AU, all these are added. Okay. Clear? Is this part is clear, guys? gRNA, it is important for you to understand. Just look at the figure once. ये सबको same है, ठीक है? So we just wanted to start like from the DNA sequence you get this, and these are the regions where U must be added. So the end product is this you are going to get. अभी कैसे हुआ है वो mechanism यहाँ पे दिया है. So you have got the unedited RNA, gRNA, they make a complementary base pairing. Then the endonuclease cut, makes a cut, adds the U and ligase reseal it. Simple. Yes. Yes. Yes, anything? Tanvi, clear? Akshaya, able to follow?
Yes, ma'am.
Okay, good guys. Yeah, so that's all. And here now I can say that the RNA splicing, the complete transcription is being done. Okay, so this is completed. And in the next session, okay, that is in the next session, we'll be starting with the translation. So what is left now is only the two topics, the translation. That also will be completing. Translation thoda sa bada hai as compared to transcription. If you could see, DNA replication, DNA damage was lengthy. Transcription chota raha, then translation is very lengthy actually, because here initiation, elongation and termination, okay, the translation of the prokaryotes and eukaryotes, hell lot of proteins are there. अभी कैसे हमने देखा था that elongation was not having those many protein factors, right? The transcription factors and all, but here we have got so many factors, the translation factors are there. Okay, so this is going to be a little lengthy. And then last, what is left is the regulation of gene. So this also we'll be doing. So these two topics are left, and then we'll be done with the mole bio, and then we'll start with the cell bio. Okay. Any doubts? Do you want to ask, guys? Tanvi, Akshay, कुछ पूछना है? What you can do is like जो भी आप videos देखते जा रहे हो, like whatever I'm uploading, even if you have got doubts in the previous lectures, and if suppose you're watching now and कोई doubt आ रहा है, so please put into the group so that we all can discuss. ठीक है? Is that okay with everyone?
Okay.
Okay, ma'am.
Yeah. चलो then. Bye bye.
Good night.
Good night.